Welcome everyone to this new um, um, new webinar of the joint collaboration between the Global Plant Council and the Plant Journal. This is the fourth and last of this season. The title of this webinar is New Insights for Plant Structural Biology, and we have three top speakers. And Katherine Denby, who will be the moderator for this session, and she's also the editor-in-chief of the Plant Journal. So a quick rehearsal of the logistics of these webinars in case this is your first, and just a quick remember if it's not your first. So as you have already started using, you are absolutely free to use the web chat in order to introduce yourselves and chat with each other. But in case you want to send a question, please use the Q&A feature tool, and we will answer all the questions at the very end of the session after our three speakers have given their talks. What else? Regarding certificates, we will send the certificates out after more or less after 7 or 10 days after finishing this webinar. So please be patient and don't overflow our emails with requests of certificates. Just be patient. If you are here, you will receive your certificate in time. Just remember that we only send the certificates to actual people and not EI note takers. That's just a reminder. So I think that's all from my side. Katherine, whenever you want, the floor is yours.
Thanks, Isabel. As Isabel said, I'm editor-in-chief of the Plant Journal, and it's great to welcome you to this webinar. We've run 4 webinars over the last 10 months, basically to provide an opportunity for authors to showcase their work and also raise awareness of special issues in the journal, which are typically collections of review articles on a key topic. So it's a real pleasure to host and introduce another 3 great speakers. The theme of the webinar is demonstrating the new biology insights and discoveries that we can make from using structural biology techniques. And the speakers will demonstrate this in different systems and processes. And all 3 speakers are also authors on articles as part of our upcoming special issue on plant structural biology. Some of the papers are already published and will be launched in the full special issue in the next couple of weeks. So look out for that. Okay, so it's a pleasure to introduce our first speaker, Clarisa Alvarez. So Clarisa is a researcher at the Center for Photosynthetic and Biochemical Studies at the University of Rosario in Argentina. And she looks at the relationship between structure and function of enzymes involved in malate utilization. And as you'll hear today, one of her key parts of her work is looking at NADP-malic enzyme and how it evolved its role in C4 photosynthesis. So I'll hand over to Clarisa, and just a final reminder, put your questions in the Q&A box at the bottom. Clarisa, over to you.
Hello.
Hello, Clarisa. We can hear you.
I'm sharing my presentation. It's okay?
Yeah, it's
okay.
Perfect.
Thank you very much. Good morning, everyone here in Argentina. I will start, if it is okay. Let me know, Isabel.
Yes, you can start.
Okay, thank you. Good morning, everyone here in Argentina, and good afternoon for you there. First of all, I would like to thank the nice introduction and all the invitation for this Plant Journal webinar series. It's very nice to be here. It's a pleasure to have the opportunity to share our work with you. Today, I will present a summary of several years of collaboratory research aiming to understanding how NADP-malic enzyme evolved into one of the key enzymes in C4 photosynthesis. Especially this structural biology work was carried out by a synergic collaboration performed with Professor Veronica Maurino at the University of Bonn. Each result that I will show you represent a huge work of many collaborators with expertise spanning biochemistry, structural biology, computational biology, and protein evolution. And I have already been introduced. I developed my biology structure work at CEFOBI Institute that is located in Rosario. And you may be wondering where Rosario it is. Rosario is a very nice sized river city located in Argentina. And the most important is the born town of Lionel Messi. And as many Argentinians, and I am passionate about sports, not only football, but also basketball. And although this topic may seem far away from my structural biology work, I will try to showing that it's the main focus of my talk. Both football and basketball share a common identity. They both use spheric balls. One is specialized to be kicked, and the other is specialized to be dribbled and passed by hand. So this difference shows that structure shapes performance. And as we will see today, evolution applies the same principle to proteins. Closely related enzymes can acquire structural changes that enable new biological functions. And this is precisely the story of C4 NADP-malic enzymes. To introduce the biological context of this work, let us briefly consider the evolution of C4 photosynthesis. Most plants perform C3 photosynthesis, in which rubisco catalyzes both carboxylation and oxygenation, reducing carboxylation fixation efficiency. C4 plants evolve a carbon concentration mechanism that elevates CO2 levels around rubisco, which is located here, minimizing photorespiration. Remarkably, this adaptation evolved independently more than 60 times, making it one of the best examples of convergent evolution. A key enzyme in this pathway is NADP-malic enzyme, which releases CO2 in the bundle-shaped chloroplast. Although NADP-malic enzyme originates before C4 photosynthesis and is widespread in plants, it was repeatedly recruited into the C4 pathway. This recurring evolutionary transition provides a unique opportunity to understand, how did an ancestral non-C4 NADP-malic enzyme evolve into a C4 specialized enzyme? How this was? Gene duplication provides the evolutionary basis of NADP-malic enzyme functional diversification. Following duplication, one lineage retained ancestral non-photosynthetic feeders, whereas the other underwent neofunctionalization and acquired C4 photosynthetic properties. In maize, the resulting C4 and non-C4 isoform still share 82% of identity. However, they have different properties. Especially, C4 enzyme form a stable tetramer, displaying higher catalytic efficiency than the non-C4 counterparts. And I will try to focus during my talk to these two main differences. Which structural changes accompany NADP-malic enzyme neofunctionalization during C4 evolution? Comparative evolutionary analysis identified two main sequence signatures: 15 additional N-terminal residues found in non-C4 isoform, and 20 different residues conserved between each isoform. This raises another question: where are these residues located in the structure, and how they contribute to oligomerization and catalytic performance? To address these two main aspects, we use an integrative structural biology approach. We combine structure and biophysical techniques, such as X-ray, crystallography, cryo-EM, extractive crystallography, cryo-EM. Also, we perform analytic ultracentrifugation and mutagenic studies. All these experimental approaches were also combined by in silico approaches, like homology modeling, evolutional hotspot analysis, molecular docking, molecular dynamics, which help us to overcome some experimentally approaches. To understand where were located the differences between C4 and non-C4 isoform, we first need to obtain the first crystal structure of a plant NADP-malic enzyme. We did this. We did it for two species, C4 photosynthetic species, maize and sorghum. The structure reveals us that it has a dimer-to-dimer conformation, similar to what was previously reported by other malic enzymes from, for example, human malic enzyme. We could detect 4 catalytic dimers and study this structure. Importantly, with these structures, we could locate the 20 residues that differentiate between C4 and non-C4 isoform. What we did then, to investigate their roles in C4 specialization, we first analyzed the oligomeric state of all these enzymes. We generate mutants of the 20 residues differentially conserved. We introduce the non-C4 residues into the tetrameric C4 background, effectively testing whether any of them change to a dimeric conformation. Of the 20 residues analyzed, we found that 16 of them now have effect on oligomerization. They conserve the tetrameric oligomerization. Only 4 of them that are marked with red stars destabilize the tetramer. What we found? We found that threonine 163 and aspartate 164 partially destabilize the tetramer, where we did the reverse mutation in the non-C4 background, where they are dimers. We tried to introduce the residues conserved in the tetrameric isoform. And we found that this promotes higher order oligomeric states. Besides, we found that the mutation of a glycine 200 promotes a complete transition to a stable dimer and to a stable tetramer in the reverse mutant. Notably, a single glycine to arginine substitution at position 200 was sufficient to switch the enzyme from a tetramer to a dimer. So, where is this glycine located within the structure, and how can a single amino acid substitution could perform such dramatic effect? To understand this, we performed cryo-EM to confirm all the oligomeric states of each protein. We confirmed that C4 was tetrameric and non-C4, and the glycine 200 was dimeric. As glycine mutation generates a tetramer to a dimer transition, we crystallize the protein. Besides all these experimental approaches, we perform also machine learning basis output analysis together with evaluation of hydrogen bonds, salt bridges, hydrophobic contacts, consistently finding that glycine 200 and threonine 163 and aspartate 164 were key interface regions controlling tetrameric stability. So where are they in the structure? Glycine 200 is located on the surface pole loop at the dimeric interface that you can see here in green. On the opposite monomer, the threonine 163 and aspartate 164 are positioned directly across this region, forming a close interface contact. This element defines a local interaction network at the dimeric interface that appears to influence higher order assembly, the tetramer. How is this performed? Aspartate 164, which we talked was involving in oligomeric conformation, is proximately to tyrosine 632 that is located at the C-terminus, suggesting a structural link between the interface region and the C-terminal domain. To test this, we generate mutants, replacing the tyrosine by phenylalanine, preserving aromatic character while disrupting hydrophobic hydrogen bonding capacity. This mutation destabilizes the tetramer state, supporting the functional relevance of this contact. Overall, these results suggest that perturbation at the dimeric interface can propagate to the C-terminal region, modulating tetrameric assembly. Altogether, these results suggest that C4 evolution was not driven by its isolated mutations, but by the emergence of coordinate interaction network that stabilized the tetramer. We propose that this quaternary state transition was a key adaptive step, improving protein stability under high cellular concentration and providing the structural context for the evolution of C4 regulatory properties. The second aspect we wanted to understand was the catalytic performance. To address this question, we decided to focus on a single plant species and compare its own malic enzyme isoforms. This was selected to avoid sequence differences arising from interspecies comparison and concentrate exclusively on the molecular determinant associated with C4 specialization. For this reason, we performed a structural study of C4 and non-C4 NADP-malic enzyme isoforms from maize. As a structural biology, one of our goals is to visualize enzymes through their catalytic cycle. However, obtaining experimental of all the relevant catalytic states is rarely straightforward. Crystal structures often capture only a subset of conformation involving in catalysis. No single technique could provide all the answers. Instead, we combine complementary structural approaches to reconstruct the snapshot along the catalytic cycle. At the time, only an apo crystal structure of maize C4 isoform was available. To overcome this limitation, we integrate crystallography with molecular docking and molecular dynamics simulations to reconstruct the catalytic relevant states. What we did, we took this structure and we performed with the product and with the substrate molecular docking and molecular dynamics. This strategy provides structural representation of the distinct catalytic states and allows us to analyze the molecular interactions associated with substrate binding and product formation. However, understanding the C4 specialization requires a direct comparison with the non-C4 counterpart. So this brought us a next challenge. The next challenge was that we didn't have the non-C4 NADP-malic enzyme structure. So we first crystallized the first non-photosynthetic plant malic enzyme structure report to date. The structure revealed a dimeric assembly, and it was co-crystallized with the proton bond state. However, as we discussed before, structural biology rarely provides every catalytic state experimentally. To characterize substrate recognition, we need a model of the enzyme in the presence of malate. In order not to use the model that is close conformation with the pyruvate as the initial state, we generate alternative structural models using homology modeling and AlphaFold models predictions. With these models, we compare with the experimental determinant crystal structure and found that homology models was structurally closer. Once we have reconstructed all the catalytic states, we could now compare C4 and non-C4 bound with malate and with pyruvate. Let's start with pyruvate, what we have found. Although pyruvate occupies a very similar position in both enzymes, the interactions around it is not identical. You can see here. In C4 enzyme, pyruvate is mainly established by lysine and tyrosine pair that was previously reported as a catalytic pair relevant of human malic enzymes, while non-C4 enzymes use a different set of interactions. Therefore, even though both enzymes bind the same product, they do so through distinct interactions. Let's now focus on malate, that is the physiologic substrate. Unlike pyruvate, where we mainly observe different interaction network, malate reveals a notably different conformation, a close conformation, as you can see here. Look a bit deeper, see that malate has in C-form isoform a linear conformation, while in non-C-form isoform is a close conformation. Okay, substrate and pyruvate, substrate and product have different binding modes in both isoforms. And what happens with cofactor coordination? I'm not going through each individual interaction shown here. We compare them, but I want just you to focus on this. Look from the transition from substrate to pyruvate in C-form isoform. It is accompanied by a marked loss of NADP interaction, as you can see here in black lines. When we look in the transition from non-C4 malate to pyruvate, it preserves mostly all NADP interaction network. Together, these results suggest that C4 evolution optimized the entire catalytic cycle, including product release. So together, all our results indicate that C4 specialization emerges through coordinate structural changes at multiple scales. Evolution optimized both oligomeric assembly and ligand coordination, giving rise to the highly efficient photosynthetic NADP-malic enzyme. In other words, structure shapes performance. Before finishing, I would like to thank all the people who contributed to this work. This project is the result of many years of collaboratory work. First, I would like to especially thank Professor Veronica Maurino for a long-standing collaboration work present today. I would also like to thank all the collaborators who contributed to this project. I will especially mention the people involved in the last part of this project, Jonas Mon and Simon Williams for protein expression and kinetic studies, Dr. Klinke for structural studies, Dr. Geyer and the crystallography facility at the University of Bonn for support in protein crystallization and X-ray data collection, and Drs. Samareño and Costabel for molecular dynamics. And finally, I would like to thank CEFOBI Institute to provide me the opportunity to develop my research in structural biology in C4 enzymes, Dr. Drinkovic and Saigo for their continuous support. Thank you for all your attention, and I will remain for any question.
Wonderful. Thank you, Clarisa. That was really fascinating insights. So just to remind people, if you've got questions, for Clarisa, put them in the question and answer, and we'll answer questions at the end. But thanks for getting us off to a great start. So our next speaker is Cecilia Blikstad Cecilia leads a research group in the Department of Chemistry in the Angstrom Laboratory at Uppsala University in Sweden. Cecilia studies the cyanobacterial carbon dioxide concentrating mechanism, carboxysomes, looking at the structure and function, assembly and regulation of these, with a main sort of, I guess, overall aim to try and, well, one aim to try and sort of inform engineering of microorganisms and crops with enhanced photosynthesis. So we're staying on the photosynthetic theme, and we'll go over to Cecilia for her presentation. Thank you.
Thank you so much. I will just share my presentation here. Okay, do you see the right view now?
Yes.
Great. Thank you so much. And first of all, thank you, Isabel and Katherine, for inviting me to give this talk. And thank you, Katherine, for this nice introduction. My name is Cecilia Blikstad, and as said, I'm a group leader here at Uppsala University. And first of all, I wanted to announce that we have an open postdoc position. So if anyone is interested, just feel free to send me an email, and I will get back to you. So I will talk about work that was started in UC Berkeley when I was in Dave Savage lab, and then I will, in the end of that will be the first part of the talk, and then I will talk about what we are doing in my lab today here in Uppsala. And the talk will focus on encapsulation and regulation of carboxysomal carbonic anhydrases. And I will, if you don't know what that is, I will tell you very soon. But I will first talk about CO2 fixation. So as all of you or most of you know, most of the CO2 fixation in the biosphere goes through the Calvin cycle and via the enzyme Rubisco. And Rubisco is actually, and Clarisa talked about Rubisco in her talk as well, and Rubisco is actually the most abundant protein on Earth, but it is famous for being a bit slow and also importantly non-specific. So it can catalyze the reaction, or its natural reaction with CO2, but it has this inherent problem that it can't distinguish between CO2 and oxygen. And if it catalyzes the reaction with oxygen, it causes photorespiration, which is a costly metabolic pathway where it has to regenerate this 2-carbon substrate or product. So plants have evolved different mechanisms to overcome this, and Clarisa talked about the C4 mechanism in plants, but in cyanobacteria, they have evolved a biophysical mechanism to avoid this instead, or to limit this photorespiration as much as possible. And what the main function of this CO2 concentrating mechanism is to elevate the concentration of CO2 around Rubisco so that it basically competitively inhibits the reaction with oxygen, and then the cell can proceed with carbon fixation in a higher rate. So in cyanobacteria, this CO2 concentrating mechanism consists of, do you see my arrow or my pointer? Maybe not, maybe I should, maybe it's okay anyway. I don't know how I get this up. Okay, you will have to bear with me, even if I can't point.
Oh yeah, we can now, that white arrow.
Do you see the white, my white arrow? Okay, perfect, thanks. Then I will. So in cyanobacteria, the CO2 concentrating mechanism consists of this energy-coupled inorganic transporters, and then a big, what we call bacterial microcompartment, or we like to call them a bacterial organelle, called carboxysomes. And these carboxysomes is what I'm going to focus on today. They are a big organelle, but it's a bacterial organelle, so they consist of a protein shell instead of a lipid shell as in eukaryotic organelles. And then they are filled with rubisco and an enzyme called carbonic anhydrase. So in the CCM, the organic transporters pump in bicarbonate into the cell. This bicarbonate diffuses into the we can do like this, into the shell of the carboxysome through pores, where the carbonic anhydrase converts it to CO2, which then is the substrate for rubisco. So these organelles are self-assembling protein organelles, and we are working on trying to understand how they assemble and how they regulate. And to do this, we use a combination of structure biology, but also biochemistry and enzymology. And in these thin section of, this is of a cyanobacterial cell, you can see these carboxysomes at these icosahedral structures. So to me, I find them super cool, these structures, which you can we can even isolate them, so we can purify carboxysomes, So if we take a more structural look on this, I should point out that this is a model. This is not the true, we haven't solved exactly how they look. So this is a model that is just built. But it has these hexameric shell proteins, which that builds up the facets. It has pentameric shell proteins, which cap the corner, and then it's filled with rubisco and carbonic anhydrase. And just for you to get a view of how densely packed these organelles actually are, I want to highlight a collaboration we had with Lauren Ann Metzgas, who was then in Grant Jensen's lab at Caltech. Now she has her own group at Purdue University. But we did cryo-electron tomography and subtomograph averaging on purified carboxysomes. So I purified the carboxysomes, and then I handed them over to Lauren Ann, and she did the cryo work on this. But what you can see here highlighted is that these dots here is actually rubisco, the one that looks like donuts. So, and in this mostly packed one, which we call ordered one, it's extremely high concentration of rubisco. So we counted this to go up to around 650 micromolar of protein within the compartment. And I don't know if someone has purified protein or something, maybe you are happy if you get out 100 micromolar protein from a normal protein prep. So this is actually very tightly packed, which I find it very cool. And we also saw that the rubiscos kind of stack together, almost like long fibrils in these mostly packed structures. So that was a little introduction, and then now I will go into first of all, tell you about why we started this, and Katherine highlighted some of this. So it's basically, first of all, to understand the basic biology of CO2 fixation in these systems, but also we believe that if we understand how this works, we can maybe improve organisms and crops. So if we understand that, we might be able to improve CO2 fixation in cyanobacteria and also engineer other industrial relevant microbes to grow on CO2. So in this paper, which is highlighted here, is a work from a graduate student in Dave Savage lab, where we reconstituted the full bacterial CCM into an E. coli, and actually he created an E. coli which could grow on CO2. Then there are also ongoing work where groups in both Australia and UK are trying to, also in US, there's a lot of groups doing this, trying to transplant carboxysomes and this transporter into C3 crops to make them grow better. And there are mathematical models showing that this can increase photosynthetic efficiently quite dramatically if we manage. And today one can basically assemble carboxysome structures, but some of the parts are still missing. Yes. So just a quick, so there are two types of carboxysomes, and we won't go into detail about this, but I would just want to highlight that there are beta carboxysomes and there are alpha carboxysomes. And the type I will talk about, focus on today, are the alpha carboxysomes. And they are found in oceanic cyanobacteria and also in some chemoautotrophic proteobacteria. And what my lab does is that we primarily study the carbonic anhydrase in these carboxysomes. And what is really important for the CCM to function is that the carbonic anhydrase is only active in the carboxysome and not in the cytosol of the cell. Because if it's active in the cytosol of the cell, the bicarbonate that's getting pumped in will get converted to CO2, and then it can escape the cell membrane again, and this short circuit the CCM. And this is also shown about 35 years ago by Price and Badger, where they expressed a human carbonic anhydrase in the cytosol and saw that created a high CO2 requirement phenotype. So there needs to be both efficient encapsulation of the carbonic anhydrase, so it actually goes into the carboxysome. It also needs to be regulated, so the activity is turned off in the cytosol and turned on when it comes to when it enters the carboxysome. So in the first part of the talk, I will talk about how we believe that this carbonic anhydrase is actually encapsulated in beta carboxysomes. So if we look at a more cartoon version of the alpha carboxysome, we have these shell proteins, and then we have a fairly big intrinsically disordered scaffolding protein called CSOS2, which connects rubisco with the shell. And as you see in this cartoon, the CA is missing. So when we started this work, we didn't know how the carbonic anhydrase actually gets into the carboxysome. But when we started to look at the sequences of these enzymes, we found that from, I think it's 300 sequences or something, that all of them have an unstructured or an intrinsically disordered N-terminal region. It spans between 30 to 130 residues, but they all have some part of this, and it had an unknown function. So my idea was, okay, this actually has to do something, otherwise it wouldn't be conserved in this way. So what we did is that we basically went fishing for what was also not known, I was jumping here, was that which protein does the CA interact with. So that was the first step. So what we did was that we kind of went fishing. So we used BLI, which is a technique called biolayer interferometry, and screened basically binding of CSOA to all the different so this was an in vitro method for all different carboxysome proteins. And what we find is that the only protein that we see an interaction with is with rubisco. What we also find is that if we remove this, which I actually don't have a picture of here, this N-terminal region of the CA, we lose the binding. So that was an indication, okay, it's actually this N-terminal region is responsible for binding to rubisco and also probably then for getting it into the carboxysome. We then turned to structural biology, to cryo-EM, to confirm this, where we were able to, with help of Thomas Loeffling and Jonathan Remis at UC Berkeley, were solved a sub-2 angstrom cryo-EM structure of a peptide of basically of the NTD in complex with rubisco. And we see that it binds at this. So rubisco is an octamer, so it has 8 large subunit and 8 small subunits. So this blue here is the small subunit, and the yellow orange is the large subunit. So we find that the Rubisco has then 8 binding sites for this peptide, and it binds in a cavity between 2 large subunit and 1 small subunit, which is probably ensures that it can only bind to fully assembled Rubisco molecules. So it can't get basically non-functional Rubisco, it will not bind to. We also find that it binds, so earlier work from the same group had shown that this scaffolding protein CSOS2 also binds to Rubisco. And we actually find that it binds to the same site, but in 2 totally different binding modes, and the peptides are very different as well, which was a surprise that they were so different and still use the same binding pocket. We were also able to confirm that in purified carboxysomes, that if we remove this NTD or parts of the NTD, this CA is not targeted into the carboxysome. Also in vivo work this is in a proteobacteria, not actually a cyanobacteria, but it's in a proteobacteria that has these carboxysomes, but we can do genetics in this one, which is why we use it as a model system. If we remove the NTD, here they don't grow, basically. So that's basically a proof of that this NTD is functional for this reason. So then we know how this, or we think we know how this enzyme is getting into carboxysomes. And the next question was, okay, how is the enzyme regulated? And our hypothesis here is that this is done by its redox states. So earlier work have shown that when the carboxysome is fully assembled, this excludes reductants. Which leads to that the carboxysome lumen becomes oxidizing, and then disulfides can form. And this has actually been shown in beta carboxysomes of this protein called CCMM. It has been shown that it's redox regulated, but in alpha carboxysomes, the mechanism for this has been unknown. It has been suggested to be redox regulated, but the mechanism for it was unknown. So, and to point out that these are 2 evolutionary very different enzymes, even if they're both carbonic anhydrases, but they come from different families. So we don't expect them to basically function in the same way. So what we did here was that my PhD student, Nicole Vogiazzi, measured steady state carbonic anhydrate kinetics. And to do, as you might know, carbonic anhydrases is one of the fastest enzymes. And so to do this, we need to use stop flow spectrometry, which is basically a technique which mixes liquids very rapidly, and then you measure kinetics. So we measure kinetics within the time frame of 0.5 seconds. Because the reaction itself is over within almost 60 seconds. So this is not within the time frame as you can mix and invert a cuvette. So we need to use this machine called stop flow. And Nicole was able to show that if she reduces the enzyme, it gets inactivated. So here is in green is the activity in oxidizing condition. In purple, we have reducing condition, and the gray one here is the background. So you see that it's basically down to 0 when we reduce the enzyme. She also showed this for a cyanobacterial version of the enzyme. We also complemented this with bioinformatics and basically showed that this mechanism seemed conserved across proteon cyanobacteria. Nicole also found a conserved cysteine pair, which is located pretty far, I think it's 35 angstrom away from the active site. where if she mutates these cysteines, we lose all activity of the enzyme. So this disulfide probably function as a regulatory switch. So then we turn to structural biology again, and with help of Guillaume Gallier, who is a researcher in my group, we solved cryo-EM structures of CSOS-CA, of this carbonic anhydrase. And we saw that it is a dimer of trimers, so it's a hexamer. However, we saw when we noticed that our hexameric map was had very clean densities in the core, but it was kind of fuzzy in the edges. So therefore, we locally refined these as dimers instead to get the maps cleaner. So what I will show you from now on will be the dimers instead of the hexamers. And we went ahead and solved, or Nicole and Guillaume went ahead and solved structures of the active oxidized protein and the inactive reduced protein, and also of the inactive cysteine mutants. And from just the first look here, they look very similar. But if we overlay this, the active version on the two inactive ones, we see that there is actually pretty large global shifts within the enzyme. And if we kind of flip in between here, we see that it seems to be moving. And what a big relief for me was that when we overlaid the two inactive versions, so both the cysteine mutants and the reduced one, we see that they almost perfectly align globally. So that was an indication that also this inactive cysteine mutants actually is resembling the reduced state. If we look closer to the actives, or we zoom into the active site, we see some critical shifts between these two, between the active and inactive states. We have a loop that's flipping. So however, we are today unsure what this loop is actually doing, but what we think is more importantly is this histidine up, which is coordinating a substrate water. And in the oxidized version, it's in a close or a near state where it can coordinate this water, which is this red dot up here. And then in the inactive states, it's been moved out and it actually doesn't coordinate the water anymore, and it can't perform the catalysis. There is much more to this story, so if you are interested, you are more than welcome to read the preprint, which is currently in revision. But for the time of it, we don't have time to go into all of the details. But with this, I want to end with that with these studies, we basically updated the model of alpha carboxysome organization, where we have that the CA interacts directly with Rubisco. We also know that this is the assembly occurs by local phase separation from this paper from Luc Oltroge. And then when the shell is fully assembled, it blocks out cellular reductants, the carboxysome matures, the lumen oxidized, disulfides form, and the CEA activates. And with this, I want to end with thanking all the people that has been involved with that in this work, primarily Nicole and Guillaume, who did the most part of this work in my lab. Also Tuer, Joel, and Thomas was involved in it. And then the people in Dave Savage lab, first of all, Dave and Eli, Thomas, Julia and Sophie, Mira and Jonathan. And then also the cryo-EM facility here in Uppsala and Stockholm, which is really great and helps us do this work. Yes, and then I want to thank you all for listening. And I'm happy to take questions after Björn's talk.
Wonderful, thank you, Cecilia. I like the picture here. I was going to say, beautiful slides, very, very lovely.
Thank you.
Great. Okay, as Cecilia said, we'll do questions at the end. So keep putting your questions in the Q&A box. So we're now going to swap from photosynthesis to hormones. And our next talk is from Bjorn Paniela Pedersen, who's a professor in the Department of Molecular Biology and Genetics at Aarhus University in Denmark. And his research focuses on plant hormone and sugar transport. And he leads the Plant Path Centre of Excellence at Aarhus, which brings together researchers from different disciplines to look at what controls plant hormone distribution. So over to you, Bjorn. Thank you.
Thank you so much, Katherine, and a pleasure to be here. Thank you, everybody, for joining. I'm going to present a little bit about our work from the center that I'm a director of. So I'm the director of a center called Plant Path, or Center for Active Transport of Plant Hormones. I want to talk about many different plant hormones today, but I'll try to stay focused on auxin for the sake of time, I think. Let's see. There we go. Okay, so I'm going to talk about auxin transport today, and I'm going to talk about how it moves from the outside of the cell into the plant cell. And so, but very briefly first, so we have this center that started about a year ago that is a joint operation between my own group here at Aarhus University in Denmark in Europe, and then also a different group at the same university, as well as a university in Brussels and a university in Würzburg in Germany. And the mission, so to speak, of the group, of the center, is to establish fundamental insights into the biophysical principles of plant hormone distribution. And we do so by combining structural biology, which is mainly what I'll talk about today, as well as membrane protein dynamics and transport kinetics, some quantitative biophysics, and then, of course, combining it all with plant physiology, in vivo phenotyping, and functional characterization. And I also just want to plug one more thing, which is a tool that we built some years ago called Namdinator, which is a tool that can fit molecular models into cryo-EM maps. It was published in 2019, and it's a very, very user-friendly tool to get those initial models for your maps. So if you're working with something, cryo-EM or crystallography, this is a possibility to use a tool such as this. Okay, so with that being said, let's move on to the introduction here. So I just want to remind everybody that what membrane proteins are. So what I'm going to talk about today is transport across membranes, and assuming you can see my arrow here, I'm seeing some nodding. Yes, okay, good. So overall, we can talk about 4 different classes of membrane proteins. You can have receptors, which will receive a signal on the outside and send the signal inside. You can have enzymes that drive some chemical reaction inside the membrane. You can have scaffold proteins that can function in various ways to link together different cells, or maybe they are scaffold proteins for cofactors driving, for instance, photosynthesis. And then you also have the transporters up here, the yellow guys, which are the ones that we work on. And here you can distinguish between channels and primary and secondary active transporters. Primary active transporters normally use chemical energy from ATP to push a substrate against the electrochemical gradient, whereas secondary active transporters use either the proton motive force or sodium gradient. And those are the ones we work on in my research group. So that's the wrong way. Let's see. Here we go. So I just want to remind everybody again that if you are working with secondary active transport, then you need to use the proton motive force if you're in plant cells. And the proton motive force consists of the difference in pH from the outside to the inside, as well as the difference in electrical charge, which is what we call the membrane potential. And if you use sort of standard values for a plant plasma membrane and plug those into the formula, then you get a delta G of about 30 kilojoules per mole for moving one proton across the membrane, which is approximately half of what you get from hydrolyzing one ATP molecule. So I just want to remind everybody that just moving one proton across the membrane actually generates enough energy to be equivalent of about the hydrolysis of one ATP molecule. Okay, good. So we can then use that energy. We can use it in Symport, where we're moving things, the substrate, into the cytosol. So in this case, it's from the outside to the inside of the cell, or it's from the inside of an organelle to the cytosol. Then, of course, we have Antiport, which is the opposite direction, where you're still moving the proton into the cytosol, but you're now moving the substrate out of the cytosol into the extracellular side. And you can have Uniport, which is also driven by the proton motive force if the substrate is charged, because if the substrate is charged, for instance, negatively charged, then the membrane potential will help to determine where the substrate is going. It's not only the chemical concentration gradient. Okay, good. So we have some plant challenges, for instance, associated with climate change. We have abiotic stressors like irradiation, drought, temperature rises, pollution, and so on and so forth. We have biotic stresses, insects, viruses, and bacteria. And what we really want to understand is how the plant can handle these stresses. So you are also well aware that plants are sessile organisms. So the way they respond to these stressors and the changes in environment is through plant hormones. One classical example is auxin, which is what I'll talk about today, and the tropisms, a very beautiful example here of phototropism, where you see a plant growing towards the light. So the plant is basically responding to the environment, and it's doing so by the use of hormones, in this case, auxin. Okay, so in a very simplified model of the world, which is not particularly true, but it's very easy to understand, you could say that auxin promotes root growth and inhibits shoot growth, and vice versa, cytokinins will inhibit root growth and promote shoot growth. So this means that if you have a stress factor from the outside, that could be the lack of water, drought, then you want to promote the amount of auxin, because that then promotes the root to grow so it can seek down and find more water. This is sort of the fundamental idea behind why we want to understand auxin and cytokinin, in this case, transport. There are many different membrane proteins that transport these hormones. We just recently wrote a review where we tried to summarize some of these. So what we're trying to show here is 4 different classical plant hormones: auxin, cytokinin, abscisic acid, and gibberellic acid. And some of the, or all the known transporters so far that we know are involved in the transport of these in Arabidopsis. And as you can see, there's quite a lot of different transport proteins, and this is one of the reasons that identifying phenotypes for various transporters has been extremely difficult in the field, because there are quite often other hormone transporters that can sort of cover, so to speak. Nevertheless, there's of course also plasmodesmata, I should say, and both the combination of plasmodesmata and this large number of transporters means that the balance of hormones in different tissues can be a very tricky thing to identify, but nevertheless, of course, it's quite important. We attack this from a structural perspective, so we're really interested in the fundamental different shapes that these transporters can take. And what I'm going to talk about today is the APC superfamily that you see here in the middle, which are membrane proteins that have 10 transmembrane helices, and they have a particular way of functioning that's called the rocking bundle model. So if we just sort of take a step back and think about polar auxin transport as we know it from the textbook, you could imagine something like this. Here you have 3 different plant cells. You have the plasma membrane here in some sort of brownish orange here. You have the apoplast on the outside. You have the cytosol in here, and we want the auxin to go in this direction across the screen. You now imagine the proton p-type ATPase using ATP to pump out protons to generate the proton motive force we talked about. So basically we have acidification of the apoplast, and we have are fairly large membrane potential positive on the outside. This means that something like auxin, which is a carboxylic acid, so it's an acid, actually can exist in an equilibrium between a protonated and a deprotonated form. And on the outside, on the apoplastic side, approximately 10% of the auxin, in this case denoted as IAA, 10% of the auxin will actually be protonated, and it can freely diffuse into the membrane, so it crosses the membrane. Once it's inside the cytosol where pH is 7, it actually becomes deprotonated in its main form. And that means that we have something that was described already many years ago by Goldsmith and others in 1974 as the ion trap mechanism. Okay, so we can actually have, and this is true for many plant hormones, we can actually have passive diffusion into and out of, mainly into cells, but potentially also out of cells. So we have that balance, we have the plasmodesmata, but then we also have another class of auxin transporters here now, which are known as the pins. The pins are special because they work on the polar side of the cell, so they're only located in one side of the cell, and the pins will then help to generate this direction of auxin transport. At the same time, we have proteins from the OX-LAX family, which are the ones I'll talk about in the rest of the talk here, which are the ones that are also responsible for moving auxin into the cell. So it turns out that this passive diffusion of auxin into the cell only account for about 25% of the total amount of auxin moved into the cell in Arabidopsis, and approximately 75% of the transport is actually mediated by the OX-LAX family. So it's a very important part of a larger picture to understand auxin transport and understand polar auxin transport. Okay, good. So let's look at some OX-LAX phenotypes. So OX1 was the first auxin importer that was identified, and it turned out to have a very strong phenotype. So it's very satisfying to work with. It generates agravitropic roots, as you can see here. So if you have the OX1 knockout that you see here at the bottom, you have gravity towards the bottom. You can see that whereas a normal root will grow down, in the case of this OX1 mutant, the root doesn't know which way is down anymore. We have another phenotype, which is the LAX3 phenotype, which is about the lateral roots, and it's a little harder to see, so we've quantified it over here. But basically, if you have the wild type Arabidopsis, it will have a certain number of lateral roots. If you knock out LAX3, the number of lateral roots go significantly down. Okay, so LAX3 is important to initiate lateral root growth by making sure that the auxin is located in the right places here. Okay, so the OX-LAX family is part of the APC superfamily, as I said earlier. And the APC superfamily is actually quite well known from a structural point of view. We have sodium glucose cotransporters, neurotransmitter sodium symporters, cation chloride cotransporters, and amino acid transporters that have all been worked with also from a structural point of view and from a biophysical point of view. It turns out that the OX-LAX family that you see up here is actually a bit of an outgroup in all of these known transporters, so they are quite unique within the APC superfamily, but nevertheless, we would expect them to function in sort of the same way. And this is sort of what we moved into in a recent publication that came out about a year ago. In the lab, we were able to purify, in this case, LAX3, the protein that's involved in lateral root initiation here, and get very nice and pure protein. The reason that we were interested in OX and LAXes actually came from an earlier study that we published in 2024, where we worked with forestry. This was a collaboration with Roy Weinstein's group, and what they were interested in is root initiation in eucalyptus trees. So what they would do is that they would cut these mature cuttings from eucalyptus, and then they would try to treat the cuttings with different synthetic auxin conjugates, including EBA, which is an auxin precursor, and then you'd screed for rooting enhancement. So a challenge when you do forestry is that when you make new cuts like this, it's very difficult to get them to actually make roots and grow. So this is a very costly thing. it turned out that if you used one of these conjugates that they had identified, which is a 4-CPA conjugate, you could actually improve this dramatically. So the industry standard right now, if you want to grow a new forest, is that you will dip all your cuttings in a solution of IBA. And that means if you do that, then approximately 25% of all your cuts will actually take root and will actually grow. So this is the industry standard for eucalyptus trees. And what we were able to show is that if you now mix EBA with this 1Q compound, we now have an 80% rooting efficiency. So we now have 4 out of 5 cuttings that actually take root and grow. So this is a dramatic improvement. And so this was a very exciting result from a sort of practical standpoint, but in the center, we were more interested in the fundamentals behind this. So what is it actually that happens with something like 4-CPA? How can 4-CPA actually have this kind of effect? So we started digging into this. And to do that, we collaborated with Ulrich Hams that you can see here, over here. So this is Uli and this is a frog. Uli is part of our center, and one of the many things that he is an expert is these xenopus oocyte expression systems. So what you do is you take these frog eggs that are approximately 1 millimeter across, and then you inject them with RNA and you wait for 4 days, and then your transporter, your membrane protein, will be expressed on the surface. You then add a radio-labeled substrate and then on the outside, and then you wait approximately 30 minutes, and then you see the import. So that looks something like this. This is a lot of work. And if we did this, we could show that LAX3 in this case actually transports auxin IAA. This is not a surprise. And we could show that it's pH dependent. So we can start to characterize LAX3, the LAX3 protein. If we do that, we can now generate a Km curve, and we can calculate that the affinity for auxin is extremely high. It's in the 200 nanomolar range. So it's in the nanomolar range, which is extremely high for transporters. We then turn to structural biology. Here we use bioreactors like this yeast bioreactor you can see here. Of course, there are challenges with small membrane proteins. It's difficult to get enough protein to make sure the sample is stable, establishing activity, which is what I just showed you, and then doing structural biology of these very small proteins. Let's see, here we go. So this is an example of what it looks like when we do cryo-EM on these things. So this is now a 47 kilodalton monomer protein embedded in a detergent micelle that we're trying to take pictures of. And I hope you can appreciate, even if you don't know much about cryo-EM, you can appreciate how all the 2D classes that you're seeing over here, how surprisingly similar they actually are. So it's telling you something about how difficult it is for the computer to actually align the different particles to identify the different views. This is one of the challenges of working with membrane proteins. This was a lot of work, but let's just skip that and just say that we solved the structure eventually. So we managed to have the first structure of an OX-LAX family member, LAX3 in this case, and it looks like this. So it is a monomer. It's divided, it has the 10 transmembrane helices, and those are divided into 2 domains. So you have a scaffold domain in orange and blue here that sort of wraps around a 4-helix bundle in green, which we call the bundle domain. And it's the bundle domain that will actually transport the substrate. We solved the structure in 3 different conformational states. And what you're seeing here at the very top is, imagine you take the same structure and now you slice it through the middle like an apple. And you're sort of looking at the binding site in the middle, like sort of looking at the core of the apple where the seeds are. So what you can see here are actually the binding site here in an inward open state, and then you can see it here in an inward semi-occluded state with IAA bound, and in a fully occluded state here with a different inhibitor called 2-NOA bound. So we can now start to see how the different substrates are actually interacting with the transporter during the transport cycle. And we can also identify what kind of changes happens. And what's really interesting here is that we can see going, remember again, this is an importer, so it's moving substrate from outside to inside. So what we can see is that the IA will bind in here, and then what will happen is that the green domain here will actually start to deconvolute slightly to release the substrate. So we can come up with a mechanism which looks something like this, and this is not particularly surprising because this is how all the APC superfamily members actually work. Nevertheless, it was really nice to see, of course, for the OXLAX family that it's the same. So what we think is happening is that IAA and a proton will bind to the central binding site. These are the states we still don't know what look like. And then we can see how the IAA is stabilized in the middle, and then these helices swing out by a very minute movement, actually only 2.5 angstroms before release of the substrate. Okay, this means that we can now talk about substrate recognition. We can see the IAA in the binding pocket in here. We can identify this histidine 247 as part of a proton donor acceptor. So this is where the protons would bind. We have another histidine over here that is probably also protonated during the transport cycle. And we have a number of other residues that we know are important for this. And that leads me to the last point of my talk here, the last minute or so, which goes back to the forestry point, right? So the whole point about the forestry was that it was not IAA, it was not auxin, but it was actually other kinds of synthetic auxin substrates that are being transported and presumably move into the whole plant, in this case in eucalyptus, to actually promote this root growth. And so you will notice down here that we have natural auxins like IAA up here. These are the ones we find in plants. But then we also have synthetic auxins here in yellow. You can see 4-CPA here and 2,4-D here, which are, as you will notice, very similar chemically speaking. And so we now know that 4-CPA can actually promote root growth, like we showed in the forestry paper. We also know that compounds like 2,4-D actually constitute approximately 20% of total worldwide herbicide usage in agriculture. So a very, very large fraction of all the herbicides used worldwide are actually synthetic auxins. So it also becomes relevant to understand how these compounds are actually penetrating when you spray them on the plant. How are they penetrating into the different tissues to have their effect? So to look at that, we basically, in the same paper, we solved structures with, besides IAA, we solved structures with 2,4-D, which was a synthetic auxin, and we can again describe the binding site. And without going into too much detail about this, also in the sake of time, we can basically show for the first time actually that LAX3 not only transports auxins, but it also transports these phenoxyacetic acids like 2,4-D. So we actually show that hormone transporters can import synthetic auxins, which was not known before. So this is sort of one step on the road to understanding how the synthetic auxins in various ways penetrate throughout the plant to have the effect that they have. And we can discuss the substrate recognition, and I'm not going to go into details about this, but I'm very happy to discuss that later if you're interested. So with that, I just want to sum up and say that some of the challenges I think worldwide in society are facing today is to ensure food security, is to ensure biodiversity, and is our consideration of how we use resources and how we handle pollution in the world. And any of these three problems are arguably manageable if you ignore the other two problems. So it's very easy to have food security if you just bomb the whole world back to the Stone Age, if you will, and sort of have fields everywhere and use chemicals on everything. Biodiversity is also very easy to solve. All you have to do is stop growing food and let nature be. And then, of course, we will all starve, but we'll have plenty of biodiversity and so on and so forth. So the challenge is to find a sustainable balance, right, where we have food security and where we still retain biodiversity and reduce pollution. And then, of course, the challenge right now is climate change, which will affect all of these problems. And we hope that in the center here, we can give some sort of biophysical foundation for understanding some of these problems and understanding how plants respond to climate change and how we can use different tools to ensure food security while retaining biodiversity. And with that, I just want to acknowledge the people who did this work. So this is Lam and Bjørn, Lynette up here. So let's just find the pointer. Lam, Bjørn, Lynette, Lorena, and Ines really did a lot of the bulk work here. And then we couldn't be here without Uli and Lucas and Sarah, who are from TUM. And then finally, Camilla and David down here really helped a lot also finalizing this story. And overall, of course, I want to acknowledge a huge number of funding bodies. I think the main thing is the infrastructure, making sure to emphasize that we couldn't do this without the infrastructure that we have access to. And with that, I think we'll stop and then get ready for questions.
Great, thank you. That's wonderful. Well, really, I mean, we've had some fascinating science talks, and what I've also liked is that they've all been linked to sort of great applications. Okay, so I will go to the question and answer. So please, if you've got questions, put them in the question and answer, and I'll go through those. I guess one question to start with to Clarisa is, would it be better for the planet if all C3 crops became C4? Are there any downsides of C4 photosynthesis, I guess?
Oh, what a difficult question to answer. No.
Can't hear you very well. I don't know if it's just me, but
Yes, it's the
Once
microphone.
again,
Yeah,
That's
that's
better.
better.
sorry about that. I was paying attention and I put. Okay, it's a difficult question to be answered. We want to introduce these residues or some of them to some type of species to allow to, for example, if you can introduce specific mutants or the different residues that we found from C4 isoform into the C3 species, like, for example, tomate, we could provide more food for more people. But no, I don't know if I'm answering to you. I don't know. We couldn't expect to introduce these changes in all the C3 species.
Okay, great, thank you. And I guess there's another question about, does mutation of specific amino acids alter the protein activity? Obviously, you've shown that is, but under different, say, physiological conditions. So you showed how the enzyme got more active, but is that always, or is it different environmental conditions or physiological conditions might impact the activity?
I didn't get the. Could you repeat, please?
So I guess you showed that you've got amino acid changes from the non-C4 to the C4 made the enzyme more active. I think this question is asking, is it always more active with those amino acid changes, or is it under specific, maybe C4 plants tend to be in, I guess, higher temperature environments? Is there a link to environment?
Yes, we are trying to do that, to link to higher temperature, for example, but we haven't tried yet. We only try these mutants at a specific pH, for example, pH 8 or 7, that we knew that they are different regulated. For example, I didn't talk in my talk because I have only 20 minutes, but we knew that C4 and non-C4 have different regulation by pH. C4 isoform is modulated by its substrate malate. It's downregulated, while non-C4 is not. So different conditions make differences with the mutants. We didn't evaluate all, but we evaluate some of them. And we have, I don't know if that's the answer for that.
Okay, so the next question, I'm going to spread them over the speakers for Clarisa. So you were talking about, say, engineering carboxysomes into plant cells, and this question was about, I guess, has there been modelling to sort of think about, I guess, it's saying the account for the size to volume differences between, say, bacteria versus a leaf, and whether carboxysomes, can still function as well, I guess.
That's a great question. I don't have the greatest answer to it, but there are not just that specific for the size, but there are modeling papers showing that if we can introduce both carboxysomes and the transporters, then probably also removing the carbonic anhydrase in the stroma of the chloroplast, we can get a quite significant increase in yield. I actually don't know how they account for the size in there, I have to admit, but there is a great modeling paper on this.
Cool. So you can go and find the answer.
Yes.
And there's another question about, you had your really nice images of the packing density.
Yes.
And just the question was, does the overall shell size vary depending on packing density, or is it regular?
in these species, or in alpha carboxysomes, it doesn't seem to do that. But it's interesting. So what's actually been seen in beta carboxysomes, which have fairly different scaffolding proteins, we see a bigger variation in size of the carboxysomes. The alpha carboxysomes seems to be pretty, like within a species, they are kind of the same size. What people have seen is that it's the length of the scaffolding protein. So if we truncate the scaffolding protein, we can get smaller carboxysomes. If we increase it, we can get bigger carboxysomes. So that seems to be part of regulating the size in some way, but not the packing, how dense they are themselves.
Not how many sort of rubiscos are in there.
No, and I mean, maybe surprisingly not, but it seems like they are kind of around 120 nanometer, like overall.
And there's another question saying, have you attempted in vitro assembly of carboxysomes?
We have. It's hard. There are probably ongoing work, but it's challenging. I mean, so what we have done and what is shown in at least one of these papers, and also work from Dave's lab, is that we can make protein condensates. So if we mix rubisco and the scaffolding protein CSOS2, the proteins basically phase separate, and we get big blobs. But these are much larger than what an actual carboxysome is. There are attempts of forming the shell around it, but we haven't seen anything that is successful yet. So that seems to be the hard part, actually forming the shell.
Right, interesting. Okay. There's a question for Bjorn about how might your structural knowledge, I guess, guide the design of molecules that can then modulate hormone transport, perhaps as a way of improving plant growth or stress tolerance?
Yes, I mean, that of course would be fantastic. And it's a difficult question, honestly, because I think from our perspective, the center just wants to understand the fundamentals of how these molecules move around. The reason it's difficult is because these molecules are acting on many different compounds. So, of course, we have the OX-LAX transporters that I spoke about here, which will import auxin. But then you need to also understand how they are exported by the PIN proteins. And you need to understand how the TIR IAA complex recognizes the auxin and how that then propagates downstream, right? So there are many different proteins that are actually recognizing these different substrates. And then, of course, you have the synthesis potentially, and you have the degradation and so on. So this is clearly a first step, but I think the forestry example really shows that there is something to be gained here. Yeah. And so, of course, the forestry example is one where we started with a list of compounds and then we just screened until we found something that worked. But our hope is that we, at some point, we can sort of flip this around and actually start some rational design where we start by understanding how things work and then saying, okay, now if we change it a little bit like this, then we actually get a new effect. And while it may sound optimistic, I think it is possible, because this is exactly what has been so successful in the past in the medical industry for various human membrane protein transporters and so on. So there's definitely a untapped potential here, I think, but it is also a difficult challenge for sure.
And I wanted to ask a question, because you had your diagram where you had the LAX promoters, LAX transporters on the left of the cell and the pins on the right. So does your structural work give you any insights into how that sort of subcellular localization is being controlled?
Yes. So first of all, the LAX proteins OX-LAX proteins are probably located all around. And so this is definitely a teaching trick to simplify the world a little bit. The pins are localized polarly, but not so for the OX-LAX proteins. I'm still trying to wrap my head around it, because there are so many different transporters that move things in, and then you have passive diffusion, and you have export, and you have degradation, and then you have stuff moving it into the vacuole for storage, and you have conjugation, and then you have breakdown. I mean, the point is, it's a very, very complex network. And I think my best guess right now is that we have to be able to model this entire network, and that requires us, if it has to be based in real biophysics, that requires us to establish some fundamental parameters of all these different transporters and processes where in this case auxin are relevant. We are very interested in the trafficking and localization of these proteins, and structural biology can help us to some extent with that, but of course there are limits to what we can talk about in that respect. I think there maybe the pins are the most interesting because they clearly have a special localization, and that must somehow be conveyed through the structure, lack of structure from the unstructured loops they have.
Right, thank you. We'll get a few more questions in. Clarisa, there's a question. Have you, with the mutating the glycine so that you make dimers, have you put that into transgenic plants and sort of investigated, does that oligomerization actually impact its role in C4 photosynthesis?
We haven't, that's an excellent question. We haven't that yet, but it's the main purpose to find out which residues we could introduce to, not to all plants compared to C4, but mostly the ones that we have we have done. We want to do this in tomato, for example, and yes, we are trying to do that.
Great. Okay, that'd be really interesting to be able to pick apart your two aspects of the enzyme. Cecilia, somebody's asked, could the structural principles that you've applied, you're sort of learning about carboxysomes, be applied to other organelles for sort of biotechnology?
Of course they could. So carboxysomes are part of this bigger group called bacterial microcompartments. So carboxysomes is one of the most well studied of these, but there are also other similar systems which encapsulate other types of enzymes. So we have the PDU compartment, for example, which encapsulate a pathway for making propanediol within cells. I think there are in E. coli and salmonella, or in salmonella, and in other organisms. So this applies, I mean, similar principles apply, I would say. And then I think this, especially, I mean, one example is this, that the interior probably gets oxidized. So it has a distinct environment from the cytosol, which is something that is probably very important to bear in mind. If you want to engineer these systems and put maybe novel enzymes in there, you have to have an enzyme which will function in that distinct environment. So I think these principles could apply to other systems. And also, if you want to engineer it, I think it's very important to understand the basic principles of how they assemble and regulate.
I guess just linked to that, there's a question coming. Do you think there's any analogy with assembly of sort of viral particles?
Viruses.
Yeah.
I mean, they look very similar, right? And that's what I normally say when I talk about them, is that they look like viruses, but they are packed with proteins instead of. But there are actually, so what people have seen is that there are no connection to viruses, surprisingly. When people have looked, especially on the shell proteins, people have done bioinformatics, and there doesn't seem to be a clear link.
Great. Okay, maybe one last question to Bjorn. There's one about, are auxins transported through plasmodesmata, and if so, is that, how sort of specific is that transport method?
That's a can of worms.
Okay, sorry. You got 1 minute.
My personal opinion is yes, I think so. It's a super complex question. First of all, you have to appreciate that plasmodesmata are not one thing, but they are many, many, many different things. So there are different types and different shapes and sizes and levels of regulation of plasmodesmata. So some of them, I'm sure, will transport auxins. Some of them for sure will not. we have no idea which one does. I mean, we can't even classify the plasmodesmata right now, so we just say it's a plasmodesmata. But so I think that's a super exciting area of research, and we are not working on it at all because that's just too difficult from a structural point of view.
Yes.
You have to address it from
Yeah.
with other methods, I think. But I would say the answer is yes and no. Sorry.
Great. Well, there's a future research niche for early upcoming researchers. Okay. Well, I think in interest of sticking to time, thank you very much to all our speakers, and thanks for the participants and your great questions. It's been really fascinating. And just to remind you about Cecilia's open postdoc position that she was advertising, and we'll be starting Plant Journal webinars again in probably September time, so look out for those, and look out for the structural biology special issue in a couple of weeks, where you can learn more about all these topics. So thanks very much, everyone.
Thank you.
Thank you. Thank
Thank
you,
you.
everyone. For attendees, you will receive an email soon with the links to the recording. And also in a few days, as I said at the beginning of the webinar, you will also receive the certificate of attendance. So just be patient, and thank you so much for staying until the very end. So thank you, everyone. Thank you to the speakers, and thank you to Katherine. See you soon.
I have to run. See you all. Have a nice day.
Have a nice.
Thanks. The same to you. Thank you again, Isabel and Katherine, for the invitation. You're welcome.
Thank you. Thank you,
Bye-bye.
Clarisa, for agreeing to do this.
Thanks all.
Bye-bye.
Bye.
Yes, thank you so
Bye-bye.
much.